Hello, it's now live. Hello, Himanshu.
Hello, sir. Yes, sir.
It's now live in Facebook. You can continue.
Okay. Okay, I can start, right?
Yes, yes.
Okay, thank you. Good evening, everyone. Welcome to this session. First of all, I would like to thank Innovation Hub Shillong Science Center and the Freebird community for giving us this opportunity. I'm Himanshu Kalita, a research intern at Margin Biologics, and we have designed some fun activities based on the NCERT biology syllabus. For today's topic, I have selected the process of osmosis. So first, I would like to demonstrate a simple experiment for a better understanding of the process osmosis. For my experiment, I have taken a raw egg. This is a normal medium-sized egg that is commonly found in every grocery store. So for our experiment to begin, I would like to weigh it in the weighing balance I have. If you don't have a weighing balance at home or near your neighborhood, it's okay. These digital weighing balance are easily available in all the grocery stores. And again, all the medium-sized eggs almost are of similar weight. They are almost 45 to 48 grams. So if you take a medium-sized egg like I have taken, it may be 45 to 48 gram in weight. So I will weight this for further confirmation. So this egg is 47 gram. So with this, now I will take another beaker and in which I will dissolve this egg in acetic acid. For the acetic acid part, I will take a commonly found item which contain large amount of acetic acid, that is vinegar. Vinegar is found in every household. As now I will I will dip this raw egg into a beaker that contain vinegar. When we dip the egg into vinegar, we can see a very clear phenomenon of appearing some water bubbles in the beaker around the egg. Let me show you. Now you can see all these water bubbles in the beaker. There are many water bubbles around the eggshell. So the reason behind is this, the eggshell contain crystals of calcium carbonate, and the acetic acid, when acetic acid that is present in the vinegar that we have put, acetic acid react with calcium carbonate and releases the carbon dioxide. All these bubbles you have seen is carbon dioxide that has been released because the acetic acid is dissolving the eggshell. That means acetic acid is dissolving the calcium carbonate that is present in the eggshell in the form of some crystals. Now this egg has to be dipped or incubated in this vinegar solution for overnight, that is for 24 hours. For that I have already dipped two another set of eggs that is of similar weight from the one I have showed you just now, and these two are I have dipped them in and these two eggs I have dipped them in vinegar for 24 hours, for almost 24 hours, and now they will look like this. After 24 hours of incubation in vinegar, the eggs look like this. As you can see, all the outer hard layer that contained the calcium carbonate is now dissolved, and now we can see the soft layer of the whole egg. I will show the raw egg also so that you can make the comparison easily. So this is the raw egg I have just dipped in the vinegar, and this is the 24-hour incubated egg in vinegar. So you can clearly see the difference between them. This has the hard white shell that we saw in common eggs, but now this one doesn't have any white shell in it, just have some little here because it is not completely dissolved yet in this part, but on the other parts it is completely dissolved, and you can see the semi-permeable membrane of the egg shell, this crystal almost crystal clear membrane. Now I will again put this in vinegar so that I can continue it continue the experiment in some other day. Now all these two egg shell which become very soft after dipping it in vinegar for almost 24 hours, we will measure this also, and we will compare the weight with the raw one that we have taken earlier. And I have told you that I have taken almost similar size of eggs, and these eggs also weigh 47 gram approximately, and I have weighed them before dipping it into vinegar. Now I will again weigh them and check the weight of it, and we will compare how the weight has changed. So for that again I will go to my weighing balance and start measuring the weight of the eggs. As our incubation period is not long enough, that is we have incubated or dipped it in only for 24 hours, there are some parts still left which is about to dissolve. If you can see, there are some calcium carbonate crystals are in my hands also because they are getting dissolved. You can see this white thing that I have sketches from the egg cell. This is calcium carbonate crystals that are present in the egg cell. This, as you can see, this is still getting dissolved, and we can just remove it by just doing this, by scratching it, but very gently because this membrane is very thin. As you can see, you almost can saw the yolk part and through this. So be very gentle with the scratching process, and you can get rid of this remaining portion of the egg cell also. And if your egg cell is not completely dissolved yet, you can try for more some amount of time, extra amount, you can dip it in vinegar, or you can increase the amount of vinegar in it, or you can increase the incubation time, that is you can keep it for 24 hours or 48 hours or 72 hours. That's depend on you, but I have kept it for only 24 hours, so there is some part is still not dissolving, but that doesn't affect my experiment, so I will weigh it. It is almost 68 gram. As you can see, this egg is almost 68 gram. Now I will take another one which is completely dissolved. Vinegar has completely dissolved its outer shell. That is the calcium carbonate present in the outer shell, and now it has become crystal clear and very soft in nature. This egg is also 65 gram approximately. As I have told you earlier that I have taken almost similar size of eggs, that is almost 47 to 48 gram. Now I have taken a egg with 48 gram. Now they have become, one is become 65, another one become 67. Why there is a change of weight in the eggs? I haven't put anything else other than vinegar. So this phenomenon is osmosis. In osmosis, water from higher concentration will go into water, or solvent from higher concentration will go into lower concentration through a semi-permeable membrane. As I have told you, this X membrane has become now semi-permeable. So water will enter from this beaker or the acetic acid part to this egg, and it will gain some weight. Now I will explain more in the process of osmosis, but for now, these are our 24 hours incubated eggs. Again, these 24-hour incubated eggs we will keep in another set of solution that we call hypertonic solution and hypotonic solution. Now, what is hypertonic solution and what is hypotonic solution, and how we can prepare them? So for hypertonic solution, it is a solution that has more amount of solute, that is solid, in compared to the normal cell. Normally, all the cell have some amount of solute, some amount of solvent, that is solid and liquid in it. But in hypertonic solution, it will have more concentration of solute, that is solid particles or molecules in it, compared to the normal cell. Then hypotonic will have less amount of solute, that is molecule or ions in it, compared to the normal cell. So for preparing a set of hypertonic and hypotonic solution, you can take any ions or molecules that will dissolve into water. In our case, with the solvent, the liquid part we have taken is tap water. But in some scientific experiments, it is depending upon the nature of experiment we are performing, or the amount of result we want from it. All the solutes and solvents are completely different from each other. So in our case, in this simple setup of experiment which we can do at home, we have taken very simple set of solute and solvent. That is solvent is tap water, that is liquid part one is tap water, that I have taken in these two beakers. And then some for the solute part, that is solid part, we have taken a very common item of our home, that is salt. In my lab, I have this bottle of salt, so I will use this, but you can use the normal household salt also. It doesn't matter. So for the hypertonic part, I will label this beaker for my convenience, so that I can remember which one contain what. This one I will prepare, in this one I will prepare the hypertonic solution. And for the hypotonic part, I will take this beaker. As I have told you that in hypertonic solution, amount of solid is more than the amount of solvent that is liquid present in it. So for the hypertonic solution, we will take a very large number of salt in it, and we will try to dissolve it very well. So I have taken a large amount of salt in this beaker that is contained the level hypertonic, and I will dissolve it very well. As you know that salt readily gets dissolved in water, so it doesn't take much time and much effort to get the salt dissolved in the water amount of tap water we have taken. So our hypertonic solution is ready with salt. It contains large amount of salt in tap water. Now for the hypotonic, I have taken the same salt but in a very low amount, so that the molecule concentration remain less and the solvent, that is liquid concentration, remain high. And again dissolve it like the previous beaker. As you have seen, I have prepared two beaker of solution. One is hypertonic that contains a lot of salt in normal tap water, and other one is hypotonic that contains a very little amount of salt in tap water. As I have told you earlier that biological system needs different type of solute and solvent. So according to the experiment we are designing, we can change the amount of solute or amount of solvent or type of solute or type of solvent. But for, as this experiment is very basic and it we can, so that we can do it at home, I have taken very commonly available items, that is tap water that is present in our home, and salt that is also present in our home, and egg cell. The reason I have taken the egg cell, because egg cell is the one cell that we can visualize through our naked eyes. We have different body cells, that is we have RBC, WBCs, but we cannot visualize them without a microscope. And as this whole activity is based on what we can do at home and at easily, so I have taken egg cell because we can visualize it that easily rather than the RBCs and WBCs. If you remember in the first part of video, I have said that calcium carbonate and acetic acid reacted, and they released the carbon dioxide gas from the egg cell, and that is the dissolving process of the whole egg cell, so that they become soft like this beakers, that is the 24 hours incubated cells. So earlier there wasn't much water bubbles or the gas release, but now as you can see there are lots of gas release in the surface of the egg cell. See, these are the carbon dioxide that has been released because of reaction of acetic acid and calcium carbonate crystals present in the egg cell. Now this cell, the acetic acid is started dissolving the calcium carbonate. After 24 hours of incubation, this cell will also become like this. The hard part of the egg cell will disappear, that is they will dissolve, and they will become very soft like this, and their semi-permeability of their cell membrane will also express. Okay, now back to the hyper and hypertonic solution. So this hypertonic solution, as I have told you, have large amount of salt. So I will put one of these eggs into this hypertonic and one will into this hypotonic, and again after 8 hours of incubation I will measure the weight, because now I have measured them and they are approximately 65 and 67 7 gram. After I will watch or I will observe what are the weight after 8 hours of incubation in this solution that contain large amount of salt, and in this solution that contain very little amount of salt. So I can't wait for this session is only 1 hour. So I have already prepared two sets of similar weight eggs that I have prepared earlier. So I have already marked this beaker also, but for convenience of the camera and lighting, I will again mark it so that you can watch it very clearly. This is hypotonic solution. That is the egg is in solution that is hypotonic, that is they have lower concentration of salt in that, and this is hypertonic, that they have a high amount of salt concentration in it. So after 24 hours of incubation in vinegar, then they will go into this hypertonic and hypotonic solution for 8 hours. As we don't have time for that to wait for this video, as the session is only for 1 hour, I have already prepared so that I can show you the final result of the whole process. So I will try to show you the hypertonic one. As you can see, this is the hypertonic. This is the egg that I have incubated in hypertonic solution for almost 8 hours. Now I will show you the egg, that is earlier stage of this egg, as they are almost similar age, similar weight. You can see this is only incubated in vinegar for 24 hours, and this is incubated in vinegar and then again in hypertonic solution for 8 hours. So you can see the difference in size. Now let us observe the difference in weight. If you remember, I have told you that I have taken almost similar weight of all the eggs. So it was almost 65 to 67 gram. Now let's watch what is the weight of this hypertonic dipped egg. So the egg was 65 to 69 gram. Now it is 68. That is, there is a slight change of weight in hypertonic solution. Now let us observe the egg that is dipped in hypotonic solution for 8 hours after dipping it in vinegar. And this is the one that I have dipped in vinegar for 24 hours, and this is the one that I have after 24 hours of vinegar incubation, it has undergo the incubation of hypotonic solution for 8 hours. As you can clearly notice the change in size of the eggs, it is become larger in size than this, and I have told you repeatedly that I have taken similar size of both similar size of all the eggs, but this you can see that this egg has become larger clearly than this or the rest of all the eggs. So let's weigh this egg and see the difference in weight. As you can see, there is a clear difference in weight. That is, it was almost 67 or 69 gram. Now it has become 77 gram. That is clear difference of almost 10 gram in this 8 hours. So I will tell you the process again so that you don't get confused. First, we have taken a raw egg, 2 raw eggs, and dip it in vinegar, that is acetic acid, for 24 hours. After dipping it in 24 hours, the 24 hours incubation in vinegar, I again prepare two solutions. One is hypertonic and another one is hypotonic with some commonly found salt and tap water. Then we have again put the eggs that is dipped in vinegar for 24 hours. Now we have put the eggs in, one in hypertonic solution, another one is hypotonic solution, and keep them for 8 hours. Then after keeping them for 8 hours, these two sets I have prepared already for you so that you can visualize the whole experiment and you can do it at home very easily. I have weighed all the eggs in every stage, and I have noticed that the raw egg has a small raw egg has less weight than the egg that is finally we are in this hypotonic stage. This hypotonic egg has solution dipped egg has the now most number the largest weight still now. So the process of the whole weight change is known as osmosis. This we can observe in another materials also, and one of the most commonly observed phenomenon is in raisins. We all have some raisins at our home, and these are some raw raisins. And now I will put this into two Petri plates. You can use any plates that you have, and hypertonic one I will. This plate has the hypertonic solution, and this is a hypotonic solution. Now I will keep this as this, and I will show this at the end of the session, and I will try to explain. Meanwhile, I will try to explain the whole phenomenon that is going on here. And for that, I would like to share a PPT with you, and I will try to explain all these osmosis, hypertonic, hypotonic, solvent, solute, all these terms that I have spoken during all my presentation, during all my experiment. I will try to explain this through a PPT that I have prepared for you. Now I will share my slide with you. I would like to request the host to allow me to share my slide with you, all of you. Sir, can you please, hello?
Okay, I'll do that right now.
Sir, please do that.
You can now share the screen.
Okay, thank you, sir. So I have told you the aim of the experiment was exosmosis versus endosmosis in plasma membrane of animal cell. Now, before explaining this term exosmosis and endosmosis, I would like to clear the confusion of solution. In my experiment, I have repeatedly said the word solution. Now, what is solution and how it is different from normal liquid we are using or a normal water we have? So solution is a homogeneous mixture of two or more substances in relative amount. That is, in solution, there has to be two parts. One is solute that I have keep repeatedly telling in my experiment, and other one is solvent. And then when we mixed both of them, they will known as solution. So our normal water or anything we have in biology or in chemistry, they are mostly solution because they are not pure thing. That is not only they don't only have this liquid part or they have this solid part. If they both if the solution if the liquid contains some amount of ions or any molecule of solid, then they will known as solution. So for solute, it always have to be some solid molecules like some iron. In our case, it is salt. You can use sugar, sugar also. Solvent is the liquid part. Most of the cases in biological experiment, we use water because it is easily available and water, everything almost dissolved in water. So solvent is always the liquid part and solute is always the solid part. In our experiment, our solvent was water, that is tap water. I will simply write water. And the solid part, that is solute part, was salt. And both salt plus water is known as solution. So I hope you have understand the concept of solution. Now I will go back to my first slide that is explaining the thing, the main part of our whole presentation and the experiment, that is osmosis. Osmosis is the movement of any solvent through from a higher concentration of solvent to lower concentration of solvent. Now I have explained what is solvent. Solvent is the liquid part of any solution. So in the process that we have called osmosis, the liquid part of a solution, it may contain some solid part also, but solid part doesn't move. Only the liquid part move through a semi-permeable membrane from a higher concentration of liquid solvent, that is where there are more amount of liquid, from to a region where there is lower concentration of solvent, that is lower amount of liquid molecules. And the whole process is known as osmosis. I will try to explain it by a simple drawing. Just imagine we have taken a beaker or anything, and now I have placed a semi-permeable membrane just in between. And now this has contained some solution. As you can see, these are the solid particle of the whole solution, and these are the solvent part, that is liquid part. As you can see, this area has more amount of solid particles than this. So if this area has more amount of solid, that it is sure that they will have less amount of liquid in it. So this has more amount of solid part, that is solute, and less amount of liquid part, that is solvent, which is exactly opposite in this right side part. Here the amount of solvent is more, that is liquid part is more, and the amount of solute, that is solid part, is less. So when osmosis occurred, this semi-permeable membrane, through this semi-permeable membrane, solvent molecule, that is the liquid molecule, will pass through this semi-permeable membrane from higher concentration of liquid to lower concentration of liquid. So the osmosis will happen in this direction. As this area has higher concentration of liquid, they will go through this semi-permeable membrane to this lower concentration of liquid. So I will change my pen color so that you can understand it clearly. Here, more amount of solvent. Here, more amount of solid particle, that is solute. So osmosis will definitely happen in this direction. That is, the water molecule or the solvent molecule, the liquid molecule will travel through this semi-permeable membrane from higher concentration of liquid to the lower concentration of liquid. This whole process is known as osmosis. Now, there is one more new thing that we are listening, that is, what is semi-permeable membrane? Semi-permeable membrane is a type of biological or synthetic membrane that will allow certain molecular ion to pass through it by any process. It may be diffusion, or it may be facilitated diffusion, or passive transport, or active transport. So semi-permeable membrane will allow, this semi-permeable membrane will allow to pass through some amount of only certain particles that they will allow. They will not allow anything to pass through it. They will allow only the liquid part or the solvent part to pass through it. There are more specific type of semi-permeable membrane. They are known as selectively permeable membrane. They will allow not only solvent but some amount of selectable molecule, that is some ions or some gases, to pass through this. So one very good example of semi-permeable membrane is egg membrane. And this is the one reason we have taken the egg cell so that the solvent part or water will easily enter into the egg cell. This is the fluid mosaic model of cell membrane, which is selectively permeable. That is, they allow not only movement of some solvent but also some gases, and that is because they have this specialized structure known as senile protein, glycoprotein, glycolipid, cholesterol. These are the reason they will act as selectively permeable membrane. They are semi-permeable also, but they are selectively permeable also. That is, they will allow both solvent and some amount of other molecule to passes through it, which will benefit for the cell. So that this specific model of cell membrane is known as fluid mosaic model. And I have already explained the solution part. Now, the endosmosis and exosmosis. So for a clear understanding of these two words, you have to clearly remember the prefixes. Endo means inside and exo means outside. So endo, if the simple way to remember or understand this is, in the endosmosis process, which is a type of osmosis, the water molecule or the solvent molecule will always enter the cell, which means the cell will have lower concentration of liquid or solvent, and the outsider environment will have higher concentration of water or solvent. So by the definition of osmosis, the higher concentration of water, it will move through the semi-permeable membrane to the lower concentration of water, and the whole process is known as endosmosis. I am saying it again. Endo means inside, that is, water always enter inside the cell. And you have seen I have placed some resins in some hypertonic and hypotonic solution. I will explain what happened to them in the last minute of the session. But endosmosis is the process in which the resins will swell when they will place in a solution, because the water from the outer environment, that I have put some water in the Petri plates, the water will enter into the cell that is inside the resin and they will swell up. This process is known as endoosmosis. Endo means entering the cell. So water entering the cell is endoosmosis. Now, the complete opposite of that phenomenon is this exosmosis. Exosmosis is mainly water moves the cell. Water moves from the cell to outwards. That is exit. Water exit the cell. This is because the water will have more concentration of liquid, that is solvent, than the outer environment. So to equalize the concentration of solvent in both cell and outside of the cell, the water from higher region of concentration, that is from inside the cell, will move to a lower region of solvent concentration, that is outside the cell, and the whole process is known as exoosmosis. Exo means outside, that is water always move outside the cell. So they are known as exosmosis. So we have learned about endosmosis and exosmosis. Endo is entering the cell, water entering the cell, and exo is water leaving the cell. But why this process is happening, and what are the condition when there is a lower amount of salt concentration in a certain cell, or lower amount of higher amount of salt concentration or sugar concentration or any concentration of solute in a particular environment? So for that, we have to learn these three new terms, that is isotonic, hypotonic, and hypertonic solutions. As you have seen, I have prepared a set of hypotonic and hypertonic solution. For hypertonic solution, I have took a large amount of salt in tap water, and for hypotonic solution, I have just take a very small amount of salt in a beaker of tap water. So hypertonic is a solution where amount of solid, I will write it down here. In hypertonic solution, amount of solid is more and amount of liquid, that is L, is less. Then in hypotonic, the amount of solid is less and the amount of liquid is more. In isotonic, there is equal amount of salt and equal amount of liquid. So when we put a cell, like I have put the egg cell in, or the resin in a hypertonic solution, the hypertonic the cell will shrink. Why? As I have told you, hypertonic solution have large amount of salt concentration or solute concentration, but very low amount of water, that is solvent concentration. So the water will move outside the cell to equalize the water concentration, that is already a higher concentration of water is present outside the cell. So what lower concentration of water is present outside the cell. As the water solution is hypertonic, they have more amount of solid in it but less amount of solvent, that is water. So the higher amount of water present inside the cell will move to outwards, and the cell will shrink, that the cell will shrink. In isotonic, this has no change, as the amount of solid and liquid is same in the isotonic. Again, in hypotonic, it will completely opposite of this hypertonic. As you have seen that in hypotonic solution, the amount of solid is less and the amount of liquid is more. Sorry, in hypotonic, the amount of so the water from outside will move into the cell, and the cell will swell up. So in hypertonic solution, the cell will shrink, and in hypotonic solution, the cell will swell up. So these are the differences of isotonic, hypotonic, and hypertonic solution. I will skip the isotonic part as it has no such role in changing the cell size or cell shape or cell weight. But in hypotonic and hypertonic, there is a change in cell shape or cell size. So for that, if you put a cell in a hypertonic solution, the cell will swell up at its swell up at it will absorb water from the outer more concentration of water environment to the lower concentration of water environment that is inside the cell, and it will swell up. But in case of hypertonic, it will release water as the outer hypertonic solution have lower amount of water concentration, and then the inside cell which have higher amount of concentration water concentration compared to the hypertonic solution. So the water will release from inside the cell to outside of the environment, that is outside solution. So that is hypertonic and that is hypotonic. Isotonic and hypotonic have not much role in any food preservation and all, but hypertonic solution has a role in food preservation. As you have known, whenever we have to store some food for longer days, like pickles and all, we put it in salt so that they will not damage for a longer period. So that is hypertonic solution do. They help in they are very helpful in food preservation because they will not allow the bacteria to grow, as there is a lower amount of water in the whole solution. So they will not allow bacteria to grow, and by the process the food will remain good or food will remain healthy for a longer period of time. That's all with osmosis. Now there's another term which is related to the term osmosis, that is osmotic pressure. What is osmotic pressure? So for the explanation of osmotic pressure, I would like to explain it through this drawing that I have already put in the slide. So as you have seen, there is a semi-permeable membrane in the beaker, and there is some more solutes in this part, right part of the beaker, and there is more amount of liquid in the left part of the beaker. So according to osmosis rule, the water will travel from left side to right side to equalize the amount of water present in the whole beaker. If we have to stop the whole process, we have to put some extra pressure from here so that the osmosis will stop. And this amount of pressure we are putting externally is known as osmotic pressure. So the external pressure which must applied, the external pressures, we must put this externally applied on the solution side in order to get the stop the flow of solvent from higher concentration of solvent to lower concentration of solvent is known as osmotic pressure. So this is the concept of osmotic pressure. With the use of osmotic pressure, we can also develop, or we can also explain the another interesting term I have put it in the slide, that is reverse osmosis. So reverse osmosis is the reverse process of osmosis. For this, I will try to explain it. Again, this is a semi-permeable membrane, and this part, the left side, have some solid molecule, that is they are more in number of solid, they have more number of solid molecule, that is solute molecule. So there is less number of water molecule, that is liquid molecule. And this side, the pure water, it has written, this side has more number of liquid molecule. So according to the law of osmosis will happen in this direction from right to left. But when we apply some external pressures, that is osmotic pressure, here the osmosis will stop. Again, if we apply more pressures, then the amount of osmotic pressure, it will forces the liquid to travel back, that is it will forces the liquid to travel back in a manner which is completely opposite to the opposite according to the law of osmosis. This whole process is known as reverse osmosis. And reverse osmosis is a very common phenomenon nowadays. We all have observed this reverse osmosis in our home regular basis. That is all our water purifiers we have. If you have noticed, all the commercial of water purifier, they mention a term Kent RO or something RO. What is that RO? That RO is reverse osmosis. This is a water purification process, and we had a water purifier we have our home is a very small example of the whole process, reverse osmosis process. For bigger plants, that is to desalinate, that is to purify the salt water, that is the sea water. There is big plants which follow the same concept or same theory of reverse osmosis to get pure water from all the salty sea water. The whole process is same, that is reverse osmosis, but they do it in a bulk manner so that we can get more amount of pure water for our daily use. As we know that there is a lot of seawater or salty water we have that we can use for our benefit, and reverse osmosis is the process that is doing the thing for us. All the country like Iran, Iraq, they have big plants of water plants. They follow the reverse osmosis process to get us a larger amount of pure water from the salty seawater. So with that, I have ended my presentation. In the whole presentation, we have talked about osmosis, endosmosis, and exosmosis. What is semi-permeable membrane, and how it is related, osmosis is related to osmotic pressure, and what is reverse osmosis and how it is different from osmosis. And also we have learned that eggshell is made up of some calcium carbonate crystals which will react with acetic acid and give rise to some carbon dioxide. Now I would like to show you the last part of my experiment that I have kept for the last minute, which is I have now I will complete the last part of my experiment. I am done with my presentation. Now I will go and finish the last part of my experiment that I have kept. As you remember, I have kept some reagents, one in hypertonic solution, some in hypertonic solution, and some in hypotonic solution. I am sure by now you have understood what is hypertonic solution and what is hypotonic solution, and why all this process of gaining some weight of these eggs or losing some weight of these eggs is happening. So now for the resin part, I would like to show you first the plate that contain the hypertonic solution. As you can see, this resin have shrink. As I explained in my presentation, if you put any cell in hypertonic solution, they will shrink because they will release their water content to the outside environment. As the outside, all these hypertonic solution have less number of water molecule in it, so the cell will release its water molecule into the outer surrounding or outer environment, and they become shrink. As you can see, these are shrinked. Now we come to my hypotonic plate. As you can see, these resins have swelled up. This resin I have put in hypotonic solution plate, and the theory I have told you, explained you earlier, that they have less amount of solid molecule in the hypotonic solution, more amount of water. So the water will enter the cell and make them swell up. So this is the summary of my whole experiment and presentation, which experiment and presentation. I hope you have understand the term osmosis and why these reagents have swelled up and these have become shrinked. All this endosmosis and exosmosis. And lastly, I would like to show you. See, there are still air bubbles are coming. That is, they are dissolving. The acetic acid is dissolving the crystal of calcium carbonate, and they are releasing the carbon dioxide gas. So there are lot of water bubble in the surface of the egg cell. The egg cell is dissolving by the acetic acid we have put in the beaker, that is the vinegar. You can take acetic acid directly also, but vinegar is commonly available form of acetic acid in household. So that's for today's experiment. In my experiment, I have explained osmosis, reverse osmosis, semi-permeable membrane, hypotonic solution, hypotonic solution, and how this works, how osmosis works, and how they help in our daily life, how reverse osmosis is very important for us. Hope you have understand, and I hope you will try this whole set of experiment, which is very easy to conduct at home. And we haven't used any exotic thing. All these things are readily available at your home, and these are very safe things, so you can try it. Please try this experiment at home and try to gain more knowledge about osmosis, reverse osmosis, and all the process involved in whole experiment. So thank you so much for today. Thank you for listening to this session. And if you have missed any step of the whole lecture or whole session, please go to our YouTube channel, Marzen Biologics. We have already put the link of the video that of this all these exciting experiments. You can watch it there again, and we have already also explained the theory behind this. So you can, if you have missed anything in this session, don't worry. Just go to our YouTube channel, Marzen Biologics. And for that's the end of today's session. And for future sessions, we will try to do more fun activities and more interesting activities. That's it for today. Thank you so much.